Good afternoon, everyone. Today I'll be discussing the contents of week 7, followed by some concepts from there and uh, some questions related to that. Let me just quickly share my screen. Okay. Please stop me in between if there are any questions. Uh, So, in the previous weeks, we have learnt a lot of basic uh, biology. What are the basic constituents in the human body? How has evolution happened? All of that. Uh, in this week, we'll be learning about immune system and various processes related to that. So we are slowly entering physiology. We have covered classification, evolution, cells, biomolecules, and cellular processes. We are now entering the realm of physiology and different organ systems associated with that. So, immune system, as we know, is the uh, system placed inside the body that prevents against any infections or pathogens. So there are the immune system can be specific or can be non-specific. It can attack any foreign body that it encounters, or it can be very specific and it will react to a specific infection that the host is going through. uh, the specific reactivity induced by host by an antigenic stimulus is known as the immune response. Now, this can be of two types: humoral mediated immunity or cell mediated immunity. The humoral mediated immunity is composed of antibody, the complement system, other components that are involved. This is mostly for viral and bacterial pathogens. Cell mediated immunity involves cells such as T and B cells. This gives protection against fungus, viruses, bacterial pathogens, and also provides immunity against cancer. There is a flip side to it. Cancer cells also have the ability to evade the immune system and slightly modify it so that they can take help from it to proliferate, but we won't be going into all of that details right now. Okay. um so we saw that antibody is coming up. So antibody is a molecule, is a complex molecule with a Y-shaped structure. These components that you see form the light chain, these two, and the others, this forms the heavy chain. There are so and these two halves are bound by disulfide bonds. This area is known as the effector region, and it is also the constant region. Like when antibodies are produced, most of these antibodies have this region constant, whereas this region is the antigen binding site. Yes, please ask your question.
Ma'am, I have one doubt regarding the assignment of week 7, in
Okay.
which the first question was the B cells are involved in which of the following type of immunity? And the options were humoral immunity, cell mediated immunity, APC immunity, and acquired immunity. So the answer is cell mediated immunity, no?
uh your deadline for the week 7 has passed, right?
Yes, ma'am. But the I have ticked the answer of cell mediated immunity in which the B cells are included. But the
Yes.
answer is said wrong, incorrect. And the accepted answer is humoral immunity. But in lecture notes, we have studied that B cells are involved in cell mediated immunity.
uh can you drop a mail to the instructor, because I don't have access to those assignment questions, nor do we set those. So maybe drop a mail to the instructor or to NPTEL.
Okay, ma'am. ठीक है.
Hmm. The division is not very clear. There is a lot of crosstalk that happens between humoral and cell mediated immunity, so maybe because of that. But you can drop a mail just to be on the clear. Okay. uh okay, where was I? So this is the constant region, uh, and this has the antigen binding site, this region. This is also known as the variable region. So the when this particularly so when the antibodies are produced, the constant regions are the one which in the gene system, they the splicing and everything goes on normally, so they are always same between the antibodies or similar between the antibodies. However, the variable region gets changed. There are different exons and introns in the genetic makeup of the variable region, because of which the cells can make different kind of antibodies and with different antigen binding sites. Okay. So when antibody is produced in the body, there are four phases that the plasma cells go through. There's the lag phase, which is the initial phase where the immune system has encountered the pathogen after its entry into the host. uh, the immune system, there will be antigen presentation, there will be a reaction of the immune system to the pathogen, but the production of antibody has not yet started, so the antibody production is usually at a baseline level at this point. Next is the log phase, where the cells that were producing the antibody know that they need to neutralize the pathogen, because of which they'll start dividing, and they'll there will be a rise in the antibody titer, and they'll start generating more antibodies. Then there will be a plateau phase, where there is a equilibrium between the antibody synthesis and the breakdown. So the plateau phase is the last stage of the infection, where the body is still fighting the pathogen, but it has started to clear off the infection. Then the decline phase, wherein there is a decline in the antibody titer. This is the phase where the infection has cleared up and the immune system can then go back to its baseline position. Okay. The production of antibody is not a single step process, it's a multi-step process. The first step is when antigen enters, of course, when the immune cells uh, encounter the antigen for the first time. These the antigen is then engulfed by macrophages, and then the antigen is processed, after which it's presented in complement with the MHC class 2. Antigen MHC2 complex is then recognized by T cell receptor, particularly helper T cell, which then signals B cells that you need to produce these antibodies against these antigen. The B cells can then mature into plasma cells or memory cells. The memory cells keep a track of what infections the body has gone through, and it is sort of like a storage that that this antigen was, the host was infected by this antigen at some point, and the memory cells the memory cells are there, so that if there is a second infection sometime later, they can quickly mount an immune response. They don't need to necessarily go through all of these processes, but the actual fighting happens by plasma cells, wherein they produce antibodies which then neutralize the antigen. Okay. The antibodies that are produced are of two types: polyclonal or monoclonal antibodies. Inside the body, it's mostly polyclonal. Monoclonal antibodies are more of a application based industrial thing, where we use the antibodies for our experiments. but inside the body whatever antibodies are produced, they can be termed as polyclonal antibodies. uh, so these but we don't, in terms of the immune system, we don't really divide between polyclonal and monoclonal. This division is essential when you are doing experiments with antibodies such as western blots or immunofluorescence assays, when you where you need an antibody against a specific protein. In those cases you consider whether you want a polyclonal or a monoclonal antibody. Polyclonal antibody, as the name suggests, is from many clones, and usually this is produced inside animals, inside mouse, rabbit, goat, etc. Monoclonal antibody comes from a single clone. It is generated by hybridoma technology, the details of which we'll go into in some time. So, once an antigen is exposed to the immune system, the plasma cells will can make antibodies against different epitopes within the antigen. So, essentially, they can bind to different areas within the antigen or different fragments of the antigen, and depending on that, the antibody is going to vary. And because against a single antigen there are so many plasma cells producing so many types of antibodies, the pool of these antibodies is known as polyclonal antibodies. Whereas in case of monoclonal antibodies, there is only a single epitope against which the plasma cells are releasing the antibodies, and therefore it is monoclonal. Was there a question? Okay. To generate a polyclonal antibody, uh, it's a very time consuming process. The first step is antigen preparation, wherein the antigen is added to a adjuvant. So the adjuvant is going to make sure that the antigen is released in smaller doses. Also, the immune system also recognizes the adjuvant as a foreign entity, but it does not have the receptors for the entity, so it mounts an immune response but does not neutralize the adjuvant. It instead tries to neutralize the antigen. If the antigen does not if the antigen can't mount a immune response, it is known as an hapten. But if you want to generate an antibody against that antigen, nonetheless, then what you do is you club the hapten with a bigger molecule such as LPS or BSA, which is going to mount an immune response for sure. Then the after mixing it with the adjuvant, the animals are immunized. It's usually done in multiple doses. Like there is an initial exposure, and then there are booster doses, and then the animal is allowed for some time to generate the antibodies, after which if there is an antibody titer or not, that's tested out using ELISA or some similar immunoassay. Then you take multiple bleeds from the animal. Now depending on the animal, if it's a rabbit versus if it's a mouse, the amount of blood that you collect and therefore the amount of antibody that you get is going to vary. But you Was there a question? uh what So the thing is, we are not really supposed to discuss the current assignment questions. We can I can do that in the next week, when week 8 starts. For week 7 I can't discuss week 7's assignment questions. I'm sorry, but that is what we have been instructed to do. But if during the class there are any doubts, I can clear that out. I hope that clears up the issue. Yes? If you have any doubts in the concepts or in the questions that I discuss, I can help with that. For the assignment questions, there is a period after which we can discuss the assignment questions, and that I don't think is over for this week. Okay. Where where was I? Huh. So you can have you can collect multiple bleeds from a single animal with some gap so that they can regenerate their blood. Then you can separate out the serum and then purify the antibody. So you will have polyclonal antibodies. To purify purify the antigen, the usually there are multiple ways to use the antigen, but using a pure antigen reduces the cross reactivity of the antibodies. uh, what some people do is they make the antigen inside, if it's if the antigen is a protein, for example, then they make the antigen inside the inside bacteria, they express it, then they isolate the protein, and then they inject the pure protein as such into the animal. That is also possible. There are two different methods: purification under native conditions and purification under denatured conditions. The purification under native conditions enables the detection of the protein at in the native condition as it is present in cell. Purification under denatured conditions helps in production of antibodies which are used for westerns or other assays in which we there is a denaturation step involved. To prepare the immunogen, you combine 100 microliter of antigen with an equal volume of Franz complete adjuvant, and this brings a final volume to 200 microliter. This is then mixed to obtain an emulsion using a syringe or a pipette, and then you can test out how good the emulsion is. Afterwards, before the initial prep of the for the initial prep of the animal, you need to take out some blood from the animal so that you have a control sample, if in case you want to test out whether the antigen has reacted in the system or not, and this is labeled as the pre-immune serum. So this blood ideally should not have any antibody against the antigen you are generating the antibody for. Then you take mice, you inject the antigen mixture around 200 microliter per mice. This can be rabbit as well, or goat, or in some cases donkey. Depends on how much volume of blood you need afterwards, and depending on that, your post processing after you have the antibody, uh, your post processing, for example, you are doing an western. In case of western, you need to after you incubate the blot with a primary antibody, you then need to incubate it with a secondary antibodies which will bind to the primary antibody and give you a signal. Depending on which animal the antibody was raised in, the secondary antibody differs. If the primary was raised in mice, for example, then we use an anti-mouse secondary. If it was raised in rabbit, we use an anti-rabbit secondary. So where the animal was raised in comes in later in the experiment. Okay. The injection can be intravenous, wherein it's injected directly into the tail vein. It can be intraperitoneal, where it is injected into the peritoneal region, but it's there should be care taken that it does not go into the stomach. Then in that case it won't produce any antibody, it will just get digested uh, and excreted out. Can be subcutaneous or intramuscular, wherein the you either in case of mice it happens in the flank region just above the leg of the mouse. After the injection, you can keep the mice back in their cage for the second booster dose, you can then combine 100 microliter of antigen with equal volume of Freund's incomplete adjuvant. So the difference between complete and incomplete is that the incomplete does not have mycobacterium shells in them, mycobacterium cell membrane. So the mycobacterium cell membrane enables a heightened immune response. If that's absent, the other components still mount an immune response, but that is not as strong as the complete one. Then you again prepare an emulsion, and then you can inject this emulsion into the mouse. This can be done for 4 to 5 times for after every 4 weeks to generate an immune response and develop memory B cells. Then you can take out some blood, try it out if there is an antibody or not, how much antibody is there. Once you know that the blood has enough antibody, then you can do the bleeding and then isolate the antibody. So next is monoclonal antibody production. The initial steps in monoclonal antibody production remains the same. Is the final steps that differ. So initially you prepare the antigen. If it's an hapten, you club it with a bigger molecule that can mount an immune response. You emulsify the antigen and you immunize the animals. After immunization, you can test out whether there is an antibody generated inside the animal or not. If the animal has generated an antibody, then spleen is taken out from the animal and B cells are, so the spleen is rich in B cells. Because of this reason, you can tweak out the spleen very slowly, and that will release B cells into the suspension. You can then use these B cells which have the memory of this antigen to create hybridoma lines. So what hybridoma is a fusion of a myeloma and a B cell. So myeloma are plasma cells which are cancerous. Because of this, they don't produce antibodies, but they are immortalized. If they fuse with the B cells, which is specific for this particular antigen, they can create a cell line which can release antibodies for this particular antigen. Then these fused lines are screened, and then the antibody is harvested out and purified. So you first culture the myeloma cell lines, you inject the antigen into the animal, you collect the spleen cells, then you fuse the myeloma cells with the spleen cells in a polyethylene glycol solution. This allows for the fusion of the plasma membrane. These cells are then allowed to grow, and if there is an antibody, if there is a antibody present, you can test out in the media that the cells in the secretome of the cells. These cells can then be screened. So each cell becomes one clone. So this is one clone, this is one clone, this is a negative cell, this is a negative cell, this is one more clone. So the cells are allowed to grow separately from a single clone, so they will produce antibody of a certain kind. Then these clones are propagated, grown in mass culture, and then they can produce antibodies. The other way to produce antibodies is you inject these cells into an animal, and then the antibody is produced in that animal. One other thing that can be done with these cells is these cells are frozen down. If required for a second or a third round of antibody production, these cells can then be revived and then used again. Okay. So this is to this is the exact procedure how hybridoma cells are made. First, you need to culture peritoneal exude cells. These are cells from bovine hypothalamus. These are feeder cells, so they are cultured with the hybridoma cells because they give nutrition to the cells, but they don't have any role in the production of the antibody. You can take it out from one mice, or if there are immortalized peritoneal exude cells, then you revive them and use them for the experiment. So this comes from the abdominal region. You expose the peritoneal cavity, inject DMEM into it, and then the cells that come out from, if you flush the peritoneum with DMEM, the cells that come out become the peritoneal exude cells. This can then be incubated with the hybridoma cells after a contamination check. To isolate the spleen cells, you make a small cut at the abdominal region after so the mice needs to be euthanized by cervical dislocation or by CO2 asphyxiation. Recent ethical recent ethics probably prefers cervical dislocation over CO2 asphyxiation, but that differs from institute to institute. Uh, so in cervical dislocation, you are essentially detaching this spinal cord from the brain, and that stops the heartbeat of the animal. CO2 asphyxiation, in CO2 asphyxiation, you place the animal in a CO2 chamber, and that because of absence of oxygen, the animal is euthanized at a later point. You remove the spleen using forceps and place it in a petri dish. You can then inject DMEM into the spleen, and this will swell the tissue. Then you can disturb the tissue little bit with the help of forceps and squeeze it out using the forceps, and this will release cells into the petri dish. You can then remove extra tissue cell clumps and centrifuge the remaining liquid. This will have the these plasma cells, which will have the antibodies against the antigen that you want to make. Then you can culture them, and so the spleen cells that you get are not immortalized. If you culture them for a really long time, they'll start dying. They won't divide essentially. Myeloma cells, on the other hand, can, because they are immortalized. So spleen and myeloma cells are mixed in a ratio of 5:1 or 10:1 depending on the experimental conditions. They are centrifuged, and then they are put in a PEG solution. Then they are put in with the PEG solution. This will allow for the plasma membrane of the spleen and the myeloma cells to fuse. The mixture is then diluted by adding some warm serum free DMEM. Uh, then the fused cells are centrifuged, resuspended, then resuspended in complete media. So FCS is fetal calf serum. It's so when you the media that we make from powders is not complete, that is called as basal media. When we supplement it with serum, bovine serum or calf serum or horse serum, then it becomes complete media. Then you add the feeder cells, the peritoneal exude cells, into 96 wells. On top of that, you add the fused cells. At this step, what you can do is you can separate out all of the fused cells into different wells, and then allow these hybridomas to grow. But then initial check by incubating the cells with the HAT media also helps, because it will kill out all of the cells which are not going to divide, which are negative cells essentially. So The hybridoma cells contain spleen cells capable of coding antibody, whereas myeloma cells does not produce antibodies and has the ability to grow indefinitely. So you are essentially taking two properties from two different lines and then fusing them. Incubating the hybridoma cells in HAT medium allows for the growth of fused cells, whereas unfused individual myeloma cells or spleen cells, not kidney cells, spleen cells does not have the ability to grow. So the myeloma cells, if grown in HAT medium, because they don't have the required pathway for assimilating the nutrients in the HAT medium, will die. The B cells, because they can't divide and proliferate, and therefore because they are not immortalized, they will undergo normal cell death. So the cells which have properties from both of these lines can grow in the HAT medium and then are considered as the hybridoma cells. So in the HAT medium has aminopterin, which prevents the de novo pathway for de novo nucleotide synthesis pathway. Instead, the spleen cells have this ability to use a salvage pathway wherein they utilize hypoxanthine from the HAT medium, because they have a HGPRT enzyme to produce guanine. Because this mechanism is absent in myeloma cells, because they are HGPRT negative, they can't proliferate. Whereas the hybrid cells, because they have properties from the B cells, they have the HGPRT mechanism in them, they can proliferate. Okay. So after you have done the initial screening, you can then expand all of these hybrid cells as single clones and then check for antibody titer and all of the properties that you, if it's reacting to the antibody antigen well or not. Okay, once colonies are observed, isolate these cells by serial dilution method, delineate the boundary of each colony, remove media to put cloning ring to each colony. You can, nowadays what people do instead is, this is also done, but another way to do this is you trypsinize the cells, you take a 380, take a very small well like a 384 well plate, and you use a cell sorter. So what the cell sorter is going to do is it can put single cells into each well. It has the ability to count single cells. So it will, instead of doing a serial dilution, what the cell sorter will do is it will put one cell into each of the wells. The process is fairly optimized, so the cell sorter, you can modulate the settings on the cell sorter to, if you want it to put two cells instead of one cell, that can also be done. But because we want a single clone, a single cell is put into each well, and then you let the cells to grow. Once the cells are confluent, you can then expand them to a bigger dish for testing. To harvest monoclonal antibodies, so the media that, if you remember from a few weeks before, we were talking about cell culture, the media has phenol red in it, because of which it will react to the pH of the pH of the surrounding. If the pH is physiological, if it's around 7, then the media stays red. If the pH is more acidic, because the cells have utilized the components of the media and then they have excreted out some stuff, the media usually becomes acidic in nature, because of which it will change to a yellow orange color. This is the sign where you need to change the media of the cells. So if the media color changes, then you can change the media into fresh media, and then the healthy cells which are producing the antibodies can be expanded. They can be put in bigger dishes to make more antibody, You can harvest the supernatant by transferring the culture into a tube and centrifuging it. Then you can add sodium. You can adjust the pH because it was at acidic pH. You can bring it to neutral pH and then add sodium azide. Then you can, for preservation, and then you can preserve the supernatant. Once you have enough supernatant, then you can do the purification of antibody. There are multiple methods. One of the method is affinity purification using an antigen coupled column. So you know what your antigen was initially, right? So you prepare a column which has the antigen lined up in the stationary phase. You equilibrate the column so that all of the buffers and everything is proper. Then you wash the to equilibrate, you wash the column twice with the equilibration buffer. Then you apply your sample to the column. You let it pass. So your sample will have multiple antibodies. If it has multiple antibodies, multiple proteins actually, the antibody against the specific antigen will get bound to the antigen. Then you wash out the extra stuff, the things that you don't need. This is essentially a receptor ligand interaction that is happening. Then you change the conditions of the buffer a little bit, and then you elute out the which disrupts the binding between the antigen and antibody. This allows for the elution of the antibody out of the column. So you inject the sample, you wash it, you then elute it at this step, after which you can see an absorbance peak in the, if you are using a spectrophotometer with the affinity column. So the antigen antibody is known as a cognate pair because the reaction is very specific. A particular antigen, a particular antibody will only bind to a specific antigen. Cross reactivity is seen if the structure is very same or the sequence is very same, but then that also means that the antigen is probably the same for both of these epitopes. The antigen antibody interactions are usually known as there is a mechanism known as a lock and key mechanism, wherein the antibody forms the lock and the antigen the key. So the antibody antigen interactions happen because there are a lot of complementary sites on both of these molecules. There are van der Waals interaction, electrostatic interactions happening. There will be certain regions within the antibody that are negatively charged, certain regions, and those complementary regions in the antigen will be positively charged. Because of these, there is a very strong interaction happening between the antibody and the antigen. Antigen antibody interactions can be checked by multiple ways. Some of these are one of these is agglutination reaction. The antigen itself is of two types, insoluble or soluble. Soluble antigens can be made into insoluble antigens with some processing. If it's a soluble antigen, you can do precipitation and then do the agglutination reaction. You can do radial diffusion assay or immunoprecipitation. We will be discussing about agglutination in a bit. Other antibody antigen interaction checking mechanisms are ELISA, Western blotting, or radioimmunoassay. All of these assays, the basis is that there is a very strong antibody antigen interaction going on. Agglutination reactions occur because it's the process of linking together antigens by antibodies and formation of visible aggregates. This, if it's an insoluble antigen, then it will involve particulate antigens such as bacteria, virus, or RBCs. These reactions are very sensitive, very reproducible, therefore can be quantified very easily. There are two types, direct agglutination and indirect agglutination reactions. Direct agglutination reactions tests it can be done against antigens such as RBCs, bacterial fungi. What usually is done is antigen and antibody are added, they form complexes, then you add anti-IgG which will form the agglutination complexes. Further on, it'll form bigger complexes, clump up the antigen antibody complexes, because of which there is a layer of antigen antibody mat formed, which will sink to the bottom of the well. If the reaction is negative, the agglutination reaction does not occur, and therefore you won't see this mat or any clumps within the micro tidal well. Okay. One of the famous agglutination reactions is used for blood group typing, wherein you test out what blood group you have using using antibodies against the specific blood groups. So the cells within the blood cells also show particular proteins against which antibodies have been raised. So in general, humans have these four blood groups: A, B, AB, or O. Uh, it can be Rh positive or Rh negative. So we essentially have 8 possible combinations. What is done usually is the blood from an individual is taken, it's incubated with anti-A and anti-B and anti-D antibodies separately in 3 different wells. If in the condition where, if there is a condition wherein there is agglutination occurring or there is clumping of blood cells happening, then that antibody signals what the blood group is going to be. For examples, if the blood is incubated with anti-A and anti-B, if there is a positive reaction for anti-A and negative for anti-B, the blood group is A. If it's the vice versa, if it's negative for anti-A and positive for anti-B, then it's a B blood group. If it's positive for both, it's AB. If it's negative for both, then it's O. So O does not have any of these antigens, A or B. This will be positive. If it's positive for anti-D, then it's Rh positive, and if it's negative, then it's Rh negative. So you take blood, you put it in 3 different tubes, you add anti-A antibody, anti-B antibody, and anti-D antibody. Then you observe what the agglutination pattern is. Okay. Indirect agglutination reactions are useful for soluble antigens because these antigens cannot go for direct agglutination reactions. They won't clump up. So what is done in this case is they are absorbed into other particles which can agglutinate, such as RBCs or latex beads. Then the antibody is against that particular antigen is added to the mixture. Then the antibody reacts, will adhere, then the particles agglutinate, and then the further processing is the same as the direct agglutination test. Okay. Let's first discuss phagocytosis before I go into the experimental part. Okay. So phagocytosis is the process by which macrophages or dendritic cells take up foreign particles into them. There are different types of phagocytosis, but the basic principle is that once the cell is in contact with the particle, if it recognizes that the particle is a foreign particle, what it is going to do is it is going to form a phagocytic cup. Depending on whether it's a receptor dependent or an independent reaction, then some downstream processes will be different. Once the phagocytic cup has formed, the cell produces pseudopode tips, Then the cell is essentially engulfing the particle. There will be endosomes reacting to the pseudopod, going to binding to the pseudopod base, and this particular complex of the complex of the particle and the cell membrane around it is known as early phagosome. Endosomes can then bind to the early phagosome, mature into lysosomes, and then release their particles. This complex of lysosome, early phagosome, and the particle is known as a phagolysosome. This phagolysosome, because it has contents from the lysosome, which are very acidic, which can denature almost everything, they can destroy the particle. The pieces of this particle can then go for antigen presentation. Depending on whether if it's receptor mediated, receptor ligand mediated, then this will be mediated by clathrin or caveolin. These are molecules that are present on the cell surface. In that case, the reaction is more active. The cell cytoskeleton is also involved in making the phagosome, but that depends on the antigen. Okay. So, how do you study phagocytosis? You can do this by either using flow cytometric methods or by using imaging methods. In imaging methods, what you study is you study the particle uptake. So, phagocytosis is essentially particles moving around in the, if there are particles moving around in the surrounding media, the cells try to take it up. So you can study it either by observing this process under a microscope or using a flow cytometric method. To observe it in a microscope, what you need is all of these materials: methanol and acetone for fixation, PBS for washing steps, mounting medium to prepare the final slide. You need something for the cells to take up. In this case, it's latex beads. So the macrophages will react to these beads. They can be of 1 micron, smaller or larger. If they are larger, the uptake will be slower. Philippine, which is a dye that reacts with cholesterol. Glass slides for doing the mounting on. Cover glasses where the cells are grown on. And all of these cover glasses, glass slides, forceps, all of these should be contamination free. J774A1 is a macrophage cell line. This can be grown in DMA media containing 10% FPS and 1% antibiotics. We usually add antibiotics into our cell culture dishes to prevent the growth of any contaminants such as bacteria. There are certain formulations where there are antimycotics or antifungals with the antibiotics as well. So antibiotics will take care of the bacterial contamination, whereas antifungals will take care of fungal contamination in the cell culture. But that's just extra information in case you need to, in case you are interested in pursuing a more practical approach for all of this. You remove the cells, remove the cells using trypsin. Then you count them, plate them on the cover glasses which were cleaned earlier. Then you incubate them to allow them for adhering. Once they have adhered, you prepare a suspension of latex beads in DMM without FPS media. So what this is just the basal media. What this will do is, because there is no serum in the media, the cells are serum starved. Because of serum starvation, they'll take up the latex beads even faster. Then you remove the media, add the latex bead suspension, then you centrifuge them if needed to let the bead settle on at the bottom. You then incubate the plate, then you wash the well, you fix it. After fixing, you stain the cells with Philippine. So what Philippine is going to do, it's going to bind to the cholesterol that is present in the plasma membrane and all the internal membranes. So if there is a membrane at any point, that's going to light up in the blue channel. Then you mount the cover slip using a glycerol or an anti-fade mounting media, and then you take it for imaging. What you will observe is something like this. So uh, if there are latex beads which are internalized into the cell, they will be internalized with a piece of the plasma membrane. In that case, you will see a blue circle around the latex bead. So in the microscope, you can locate where the latex beads are, and then see if there is a halo around it with a blue outline. In that case, the beads have been internalized using a using phagocytosis. Okay. For example, if you want to check it using flow cytometric methods, in that case, what you do is you incubate the cells with the thing that you want to be phagocytosed. In this, it can be a FITC labeled compound, it can be a FITC labeled bacteria. If it's a FITC labeled anything, you can use trypan blue to quench it. What it will do is it will stop the fluorescence from happening if it's outside of the cell. Trypan blue is a charged dye. It won't enter live cells. It will only enter dead cells. If the cell has holes in it, but in all of these processing, the cell membrane is kept intact. So FITC level bacteria which are bound to the surface of the cell or outside of the cell are going to be quenched by trypan blue. The ones that are inside, however, have fluorescence in it. You then run this solution in this suspension of cells through the FACS sorter or FACS analyzer. In any FACS experiment, you need an untreated sample, which is this, which will have very basal level of fluorescence. This is without the blue line is without any trypan blue quenching, and the green one is with trypan blue quenching. So you get a proportion of how many cells have the cell, how many bacteria the cells have taken up, and how many cells have taken up the bacteria. Both of these parameters you can know. Okay. If you want to see formation of phagolysosome into the procedure that we did for observing phagocytosis, you add an extra step of rhodamine dextrin. And you can, if you want to observe it against a specific antigen, then you can use a primary antibody against that particular protein. So again, you culture the cells, you treat them with latex beads, then you fix them. All of this is the same. However, at one point, you add the philippine and everything. To label the lysosome while the cells are growing, you grow them with rhodamine dextrin. Rhodamine dextrin will specifically go into the lysosomal compartment because it has dextrin in it, which needs to be broken down for the cells to use up. Then you wash the cells, grow it in rhodamine media without rhodamine. So you will have populations of phagosomes which don't have rhodamine, populations of phagosomes which have rhodamine. If they have the rhodamine, that means that they have fused with phagolysosomes. You can then stain them, and what you will observe is something like this. You're going to observe the bright field to locate where the beads are. You are going to observe the blue channel to locate the cholesterol molecules because you have added philippine into the mixture, and then you will observe the red channel to observe the rhodamine dextrin. So you can observe where the particle is. You can see if there is a uh, blue sphere around that particle, and if there is a red sphere around that particle. If the particle is lined by both blue and red spheres, then it implies that it has reached a phagolysosome stage. Any questions? Any questions pertaining to the contents till now? Okay. Um, Different organs within the body have different compositions, and there is some of these organs can regenerate, some of these can't, and there is a constant renewal death cycle that's happening. The in the organs where they can't undergo regeneration, they can and if they die a lot, then the organ system is going to fail at one point. uh, but because the organ systems are different, they are composed of different cell types: RBCs, WBC, lymphocytes, neutrophils, basophils, eosinophils, platelets. All of this make up the blood compartment of the body. Liver is made up of hepatocytes, Kupffer cells, and blood. Brain is made up of neurons, astrocytes, glial cells. Heart is made up of cardiomyocyte, cardiac pacemaker cells, smooth muscle cells, endothelial cells, etc. So the cell, depending on what metabolic reactions is going on, what is the density, what is the size, what are the receptors on the cell surface, uh, can go in different pathways. There is a very large variation in the cell size even within a single individual. It can range for around 3 micron to 40 microns, depends on the function of the cell. So what you can do is you can pass these cells, this mixture, if there you have a mixture of cells, you can pass this along a cell analyzer or a flow cytometer, and what you will get is there are detectors for size, there are detectors for density. If your cells are fluorescently tagged, there are detectors for fluorescence, and there is a cell collector if you want to collect these cells. Some, if it's just a analyzer, it might not have a cell collector. If it's a flow sorter, it will definitely have a cell collector. Depending on if you have tagged your cells with any fluorescent dyes or any optical dyes, then you can separate out your cells depending on that, or you can separate it out depending on the size or the granularity of the cells. So the instrumentation is looks something like this. There is a channel wherein you apply a sample. There's a laser that scans each of the cells, and if you have any fluorescent tags, or if it's just so SSC means side scatter, FL2 FL1 are fluorescent channels, and uh, FSC is forward side scatter. So depending on the on what you are looking at, you can select any of these channels, and the laser will shine these lasers on the laser module will shine these lasers on the sample, then sort it accordingly. If the cell is positive, it will be collected. If the cell is negative, you can either collect it or discard it. There is a ADC module to analyze all of this data, and then you can do further analysis on the computer. So there is a flow system for fluidics, there is an optical system that allows you to visualize the cells, and there is a electronic system, which is the computer system the flow system is attached to. The fluid system, the fluidics, has a central channel or core through which the sample is injected. There is an outer sheath which contains faster flowing fluid, uh, enclosing the central core, so that the uh, hydrodynamics of the flow cell is optimum. The optical system is a laser and an argon lamp. It can be argon lasers, or it can be an arc lamp. The arc lamp will is useful for forward and side scatter specifically. Okay. uh, there, as I said, there are different detectors, forward scatter channel and side scatter channel. Together they can give you an idea of how the cell looks like. If the forward scatter channel is high, that implies the cell size, the refractive index is more. If the side scatter is high, then the amount of granularity in the cell is more. So if the cytosolic structure is very convoluted, you will get a higher signal in the side scatter. The electronic system converts the photons that you obtain into photoelectrons and measures the amplitude, the pulse, and then processes it to show us how the population looks like. You can have different kinds of plot. There can be a single color histogram, there will be a two color dot plot, contour plot, or density plot. I am not I can mention how all of these look like in the next class. So if you want to not only analyze the data, but you want to see that if there is a specific subset, and if you want to collect them, you do gating within the flow setup. What this does is it selects only a certain population of cells for analysis or for collection. If you Usually the gating is done using an untreated sample, and then you see how the treatment changes the population dynamics. So the gating can be rectangular, elliptical, polygonal, quadrant, anything. This depends on what population of cells you want to focus on. So you can have different kinds of dyes, and all of these dyes will have different emission and excitation wavelengths. Some of these dyes are very close by, so you can use alternate channels if you don't have that specific channel in your system. Okay. So the cells that are present in a given system can be either growing very happily, or they can be stressed like in a human. So if they are growing, there is an increase in number. If they are stressed, they can either repair the stress and go into growth phase. If they are too stressed, then they either go into autophagy, apoptosis, or necrosis. Autophagy is self-eating of the cell. So the cell tries to repair the damage by eating parts of itself, the damaged parts, in the hope that it can go back to a growth phase. If autophagy fails, the cell can go into apoptotic phase. So apoptosis is also known as programmed cell death, wherein the cells have machinery in place in case the cell needs to stop itself, then it enters the programmed cell death pathway. If there is stress because of injury, the cells usually undergo necrosis and not apoptosis. There are differences between both. Okay, let me first explain apoptosis okay. So what happens in apoptosis is there are two pathways, extrinsic or intrinsic pathway, depending on what kind of stressors are there. Intrinsic pathway usually happens because of DNA damage, ER stress, hypoxia, or metabolic stresses. Extrinsic pathway are usually signals from outside of the cell. If something outside is signaling the cell to die, and depending on whether the cell is undergoing apoptosis to extrinsic or intrinsic pathway, it activates different caspases in different initiator caspases. Caspases are known as initiator caspases. There are certain molecules such as BCL2, BCLXL, MCL1. These molecules prevent the start of the apoptotic pathway. But if the cell can't stop the apoptosis, if it has to apoptosize, then these caspase 8 and 10 in case of extrinsic pathway and caspase 9 in case of intrinsic pathway, then signal effector caspases, which are caspase 3 and 7. These come later in the pathway, and these are like final signals for the cells to undergo apoptosis. Once the cell has decided, yeah, I'll undergo apoptosis, what happens afterwards? What happens is the cell starts shrinking. There is apoptosome formation, there is membrane blebbing, so the membrane starts to remodel itself and starts to engulf certain parts of the cell. The nucleus collapses. The nucleus also comes into one of the membrane blebs, and there is formation of, and then the cell disintegrates. There is formation of apoptotic body. These apoptotic bodies can be taken up by macrophages and lysed. But if there is necrosis, what happens in necrosis is there is a sudden insult to the cell. Because of this, the cell membrane itself breaks down. The cellular constituents spill out of the cell. In case of apoptosis, the apoptotic body formation prevents the contents of the cell from spilling out. There are contents inside the cell such as lysosomes and other cells, other cytoplasmic proteins. These proteins, if they are exposed to the outside, exposed to the tissue, they can cause inflammation. So in case of apoptosis, because they are covered in apoptotic body, there is no inflammation. Whereas in case of necrosis, because the contents of the cytoplasm are spilling out, there is a lot of inflammation in that area. Okay. So now you know what's the difference between apoptosis and necrosis, but the end result is the death of the cell. How do you know if your cells are healthy or if they are dying? You do an assay known as trypan blue exclusion assay. So trypan blue, as I said, is a charged dye. It can only enter dead cells. It can't enter live cells because there is absence of membrane potential in the dead cells. If a cell is dead, it can enter it, then stain it blue. So if you can use an hemocytometer or even a normal dish after addition of trypan blue to see if your cells are blue or if they haven't taken up trypan blue. To check experimentally, what you can do is you can have a hemocytometer, you can have your cells, you remove the cells from the cell culture plate by trypsinization, you plate a small amount of cells on the glass side, cover them with cover slip, then you dilute the cell suspension with the trypan blue solution. This also enables you to count less. You can then approximate later. You fill the hemocytometer chamber. This dilution of 50 microliter plus 50 microliter can be changed. If your cell suspension is very dense, then instead of this 50 microliter, you take 10 microliter of cell suspension and 40 microliter of PBS, and add 50 microliter of trypan blue into that. So that is, you get a hang of that once you culture cells, but the end result is you can observe the viable cells which are colorless and the dead cells which are blue or dark colored. You place after you place it in the hemocytometer chamber. The hemocytometer chamber here has a grid like this. So it has a grid like this. What you do is you count whatever cells are here in these squares. Then you can use the formula specific to the hemocytometer, depending on the meniscus that forms between the cover slip and the hemocytometer. You can use the formula and calculate how many cells are there as well. Okay. You can using flow cytometric methods and other methods, you can also check what cycle of the cell cycle what stage of the cell cycle cells are in. So if you remember, there are 4 stages: G1, S, G2 and M phase. There are microscopy techniques wherein you can observe where the cell is. You can also fix the cells, then stain it with specific molecules that are present in all of these in specific stages. That will tell you how many cells are present in G1 stage. If you are staining for something specific in S phase, it will tell you how many cells are present in S phase. But you can also do flow cytometric analysis, wherein if you stain the if you pass the cells through a flow cytometer uh, in an untreated sample, you see that a lot of cells are in G1, G2. You see there is a lot of there are a lot of cells in S phase, there are a lot of cells in M phase. uh, Depending on your gating, if you want to separate out certain cells, you can also do that. Okay. um okay. Now, coming, if you want to see if your cells are apoptosizing or not using flow cytometer, what you can do is you can check for DNA and phosphatidylserine. DNA can be checked using PI or probodium iodide. What probodium iodide does is, that's also a dye which can't enter live cells. It can only enter dead cells. Phosphatidylserine is one of the lipids that is present in the inner leaflet if the cell is live. If the cell is dead, the plasma membrane flips, because of which phosphatidylserine will is exposed outside. Annexin 5 is the dye that binds to phosphatidylserine, that also does not enter live cells. If the plasma membrane is flipped, the cell is dead, then only this will uh, react to the phosphatidylserine. So what you do is you stain your cells with PI, with propydium iodide and annexin 5, and then you pass the cell suspension in a flow cytometer. If your cells are here in this quadrant, that implies your cells are healthy. They don't have propydium iodide staining, they don't have annexin 5 staining. If they are here where they have annexin 5 staining but they don't have PI staining, they are starting to undergo apoptosis, but they haven't um reached the late apoptotic stage. So these are early. The ones in this quadrant are early apoptotic cells. The ones in this quadrant are late apoptotic cells because they have very high PI and very high annexin 5. The ones here have very high PI but not enough annexin 5 because the plasma membrane starts to disintegrate. So these are essentially dead cells or debris. Okay. So if phagosomes, endosomes, all of the endosomes, lysosomes, all of these uh, um, all of these compartments constitute the vesicular trafficking system. Apart from this, there are other or other pathways inside the cell that use the vesicular transport system, wherein they use vesicular transport to transfer molecules from one uh, organelle to another. So the main function of the vesicular trafficking system is to distribute the material, proteins, food throughout the cell. Majorly 3 organs are involved: endoplasmic reticulum, Golgi apparatus and lysosome. All of these together coordinate their functions to make sure that the cell is healthy. So eukaryotic cells take up solid material from outside the cell using a process called endocytosis. If they are taking liquid material, then it's called through a process called pinocytosis. If they are secreting something out of the cell, then it's known as exocytosis. These can be secretory granules, these can be excreta that the cell does not want to keep within itself, anything. So the intravesicular system delivers protein synthesized in the endoplasmic reticulum in two different organelles. It also makes sure that endocytosis, pinocytosis, and exocytosis are properly mediated. Was there a question? So during endocytosis, material that is present outside of the cell is taken up by the cell. The endosome then fuses with a lysosome to form a late endosome. This late endosome lysosome complex can then degrade the material that the cell has taken up and release the nutrients for further uptake. This endosome lysosome complex arises initially from the Golgi complex. The in case of exocytosis, if the cell wants to secrete something out of the uh, out of secrete something out, then the Golgi creates a secretory granule which matures, reaches the plasma membrane and fuses with it, in short releasing the contents inside the vesicle to the outside. Okay, during endocytosis, materials present outside binds to the cell surface through the receptors and trapped in a membranous structure called lysosome. Endosomal vesicles fuse with lysosomes to form late endosome. In late endosome, with the help of lysosomal enzymes, the material is digested and the endosome is fused with Golgi bodies to deliver content for further distribution. In a similar manner, during secretion, vesicles originate from Golgi bodies and fuse with plasma membrane to release the contents outside of the cell. Okay. So, as I said, there are three molecules involved in this. 1 second. Sorry. There are three individual parties involved in this: endoplasmic reticulum, Golgi, and lysosome. Let's first see what happens in endoplasmic reticulum. Uh, as you know, endoplasmic reticulum can be smooth or rough. The rough endoplasmic reticulum has ribosome attached to it, because of which it has a rough appearance, and here a lot of protein synthesis happens. So the protein synthesis that happens in RER can be sorted into three categories: integral membrane proteins, proteins for secretion, and protein destined for different organelles. And all of these proteins are synthesized with the N signal peptide. This peptide is specific for specific organelles. The N signal peptide are recognized by the signal recognition particle on the target organelle. If the endoplasmic reticulum is making something for the mitochondria, then it will have an N signal peptide specific for mitochondria. There will be some post processing to this protein after it's synthesized at the RER, but still the tag remains. If there is no N signal peptide, then the protein remains in the cytosol. So, functions of endoplasmic reticulum: synthesis of steroid hormone in gonad cells. This is more of a function of smooth endoplasmic reticulum. Detoxification, calcium ion sequestration, synthesis of protein, phospholipids, and carbohydrates. Phospholipid synthesis happens more in smooth endoplasmic reticulum than in rough endoplasmic reticulum. Protein sorting to different organelles, protein modification such as glycosylation. Golgi bodies first visualized by Camillo Golgi. It's essentially a stack of flattened disc-like cisternae, and there are three distinct zones. Cis phase receives the material from the endoplasmic reticulum. Medial Golgi is the place where the actual processing happens, and trans Golgi forms the vesicle for shipping out into the destined organelles or plasma membrane. So Golgi bodies are responsible for protein sorting at the trans Golgi, protein modification at the glycosyl in the medial cisternae, and proteolysis by formation of endosomes. Lysosomes were discovered by DTU. These are essentially known as suicide bags of the cells because of their role in autophagy, where they where the if the lysosomes fuse with something, then they degrade it. So the lysosomal lumen is extremely acidic and contains proteases, cytolytic enzymes, and that enable it to degrade the ingested material. Functions of lysosome include degradation of ingested food material for delivery through the vesicular system, degradation of pathogenic bacteria, and degradation of old protein. In case of cells such as macrophages and dendritic cells, the lysosome, after degrading the pathogenic bacteria, then do antigen presentation. Okay. In a lot of proteins, the trafficking signal, the where the protein should go, is glycosylation. So what glycosylation essentially means is you take the protein in its bare form, as it was synthesized in the uh, ribosomal machinery, and you, as it was translated, and you do certain modifications to it. If it's a glycosylated protein, then these modifications include addition of different sugar molecules onto the amino acids. So approximately 50% of the characterized proteins go through this alteration of glycosylation, which involves covalent addition of sugar moieties to specific amino acids. Soluble and membrane bound proteins expressed in endoplasmic reticulum undergo glycosylation. All secreted proteins, surface ligands, surface receptors ligands, all of these also undergo glycosylation. Okay. There are different types of glycosylations that happen. N and O are more prominent ones. There is also C-linked glycosylation and phosphoglycosylation happen. Coming to N-linked glycosylation, in this the glycans are covalently bound to the carboxyamido nitrogen on asparagine, therefore N glycosylation. It's the most common type of glycosylation. 90% of glycoproteins are N-glycosylated. It happens as soon as the protein is synthesized, almost simultaneously with the translation process. This happens in eukaryotic as well as prokaryotic cells. There are different events. First one is precursor glycan assembly, just after the protein has been synthesized. Then there is attachment, then there is trimming and maturation. All of the initial steps are identical for all proteins, but the trimming and maturation parts are different. So the precursor glycan assembly, the purpose of this is to assemble 14 sugar molecules which consist of 3 glucose, 2 N-acetylglucosamine and 9 mannose sugar molecules on the ER membrane via dolicol. What dolicol is a polyisoprenoid lipid carrier rooted in the ER membrane via a pyrophosphate linkage. So first 7 molecules obtained from the sugar nucleotides in the cytoplasm are added. Then after that, 7 more sugars are added to form this precursor glycan. After precursor glycan, there is after the pre-assembled glycan is attached to the newly formed polypeptide chain via enzyme oligosaccharide transferase. So this particular enzyme looks for asparagine, X, threonine or serine, this particular moiety in the polypeptide chain. If there is such a moiety found, then the pre-assembled glycan can attach to that position. Once it is recognized, a 14-mer precursor glycan is attached to the asparagine molecule, asparagine moiety, and then the beta OH group of the serine threonine residue act as the hydrogen bond donor for the reaction. So this is how it happens. In the ER lumen, after the formation of the protein, the ER lumen has these glycan assembled glycan assembly molecules, then which can then gets transferred to the protein polypeptide sequence. Sugar molecules are trimmed off by glycosidases via hydrolysis. In this process, Golgi and ER both serve different purposes. Different kinds of trimming occurs at different places. In ER, trimming is done to both protein. So there is also this thing that if the protein is not properly made or glycosylated, it won't fold properly. If it won't fold properly, it can't function properly, because a lot of the interactions are based on structural structures between two molecules rather than their specific uh, sequences. So if they can't fold properly, they can't function properly. So when the molecule is made in the ER and the initial glycosylation is done, if the molecule folds properly, then only it gets goes into the Golgi for further processing. Otherwise, the ER tries to repair the damage or sends it for the unfolded protein response pathway. I am not sure if we'll go through that or not. There is a continuous cycle of removal and addition of terminal glucose molecules that enables proper folding. At first, two terminal glucose are removed, followed by binding of calnexin and calreticulin to the chain via remaining glucose molecules. These then act as chaperones for folding of proteins. Final glucose is hydrolyzed by glycosidase 2 to discharge glycoprotein from chaperones. Folded output from this process is not yet acquired the native conformation. Now UDP glucose glycoprotein glucosyl transferase acts on non-native folded protein and transfers a glucose into the glycoprotein. So, and there are other steps so in which you can go through, but in general there is a lot of addition and trimming going on at the on the protein molecule. Then after the trimming is done in Golgi bodies, both trimming and addition of sugar molecules takes place to generate a diversity of glycan structures in glycoproteins. Depending on what state the cell is in, similar proteins can also have very different glycosylations on themselves. In Golgi bodies, each cystine carries a specific enzyme for stepwise processing. It can be manocytases, it can be glucanac transferases, and in the end, what you will end up is something like this: a protein molecule attached to a series of sugar molecules. Then there is O-glycosylation, wherein this is more common for high molecular weight proteins, especially found in mucus secretions and proteoglycan core proteins that form the extracellular matrix. These are mostly secreted proteins. It is also common in antibodies, which again is secreted. O-glycosylation does not happen instead of N-glycosylation. It happens in addition to N-glycosylation. So N-glycosylation first happens and then O-glycosylation happens on these proteins. This is carried out by N-acetyl galactosamine transferase, which transfers one single GalNAc residue to the beta OH group of a serine or threonine. Some proteins are glycosylated with GalNAc, some with glucose, xylose and mannose and so on, but the addition occurs in that beta OH group of serine or threonine. O-glycosylation forms less complex structures compared to N-glycosylation, but that happens after N-glycosylation has happened. Phosphoglycosylation is not found in humans. It is, however, found in Leishmania and Trypanosoma, slime mold, Strictiostridium. It is used by the organism to protect it from the host defense mechanism. These are also linked to serine and threonine residues by a phosphodiester bond. C-glycosylation or mannosylation are is another type wherein the glycosylation happens to the carbon via carbon-carbon bonding, not to the nitrogen or oxygen molecule. This does not happen anywhere. This has been spotted in cell lines and at liver microsomes. Okay, so this is how the vesicular trafficking pathway looks like. Are there any questions at this point? Are there any questions? Okay. Okay. If there are no questions, I will discuss some old assignment questions pertaining to this. Okay. The first question is, the cells that are considered part of the immune system surveillance system are neurons and muscle cells, monocytes, macrophages, dendritic cells, or red blood cells. Just a second. It's monocytes, macrophages, and dendritic cells. Antibodies neutralize the pathogens by binding to the pathogen to prevent its movement or function, enhancing DNA synthesis in the pathogens, destroying pathogens through lysosomal activity, or producing reactive oxygen species. The answer is A, binding to the pathogen to prevent its movement. Then other cells can attack the pathogen and neutralize it. Role of adjuvants in immunization is neutralizing antibodies, suppressing excessive immune activation, enhancing the immune response by slowing antigen release, or eliminating memory B cells. The answer is enhancing immune response by slowing antigen release. Immune cells that produce antibodies, plasma cells, T cells, monocytes, or dendritic cells. The answer is plasma cells. T cells help in this process. They also have some cytotoxic activity, but that is not mediated through antibodies. So T cells, like B cells, have memory and plasma cells. T cells can mature into helper cells or cytotoxic cells. Cytotoxic cells release molecules that affect the immune response, whereas helper cells, uh, the T helper cells help the B cells to mature. Monocytes are also immune cells, but they are a precursor of macrophages. Monocytes can, depending on the situation, differentiate into macrophages. Dendritic cells are another type of antigen presenting cells. They are also phagocytose and then present the antigen. The cells that are fused in hybridoma technology to create hybridomas are T cells and macrophages, antigen presenting cells and plasma cells, red blood cells and fibroblasts, or B cells and myeloma cells. The answer is B cells and myeloma cells. The role of polyethylene glycol in hybridoma technology is to lyse cell membranes to promote antigen antibody binding, to inhibit DNA synthesis, or to act as a fusion agent to combine cell membranes. The answer is D, to act as a fusion agent to combine the cell membrane. In hybridoma technology, myeloma cells are used to produce high level of antibodies. They provide immortal growth characteristics to hybridoma. They have the ability to undergo mitosis. They regulate cytokine production. The answer is they provide immortal growth characteristic to hybridomas. The purpose of HAT, hypoxanthine aminopterin thymidine media, in hybridoma selection is to stimulate antibody production in B cells, enhance growth of myeloma cells, allow only fused hybrid cells to survive, or to prevent antigen degeneration. The answer is it allows only fused hybrid cells to survive. Technique that uses antibodies to identify specific antigens in a sample is polymerase chain reaction, Western blotting, DNA sequencing, or cell culture. The answer is Western blotting. Soluble antigens that are typically detected in immunoassays are through precipitation or ELISA techniques, by spectrophotometric analysis, through electrophoresis, or using direct agglutination reactions. Usually that's sorry done by precipitation or ELISA techniques. If they're getting precipitated or if they're adsorbed onto some molecule that can undergo direct agglutination, then we can go perform an agglutination reactions. Otherwise, you do precipitation, ELISA, Western blot, something of that sort. Diagnostic application that involves use of agglutination reactions is RNA quantification, DNA replication analysis, blood typing, or genetic sequencing. Blood typing. The antibodies ideal for diagnostic applications, as they form irreversible covalent bonds with antigens. They exhibit high specificity and affinity for antigens. They are non-protein molecules, or they can catalyze chemical reactions. The answer is they exhibit high specificity and affinity for antigens, but they do not form irreversible covalent bonds. They mostly react by non-covalent bonding. The process which ensures that the apoptotic cell debris does not cause inflammation is lysosomal degradation, engulfment of the apoptotic bodies by macrophages, release of cytokines, formation of necrotic vacuoles. The answer is engulfment of apoptotic bodies by macrophages. The dye that is commonly used to stain cholesterol during phagocytosis is rhodamine, philippine, uh, trypan blue, or FITC. The answer is philippine. Rhodamine is used to stain the lysosomal compartment. Trypan blue will stain the dead cells. FITC is just a dye which can be bound to anything. The double fluorescence ring, in case of rhodamine, so this rhodamine should be rhodamine dextrin. If it's just rhodamine, it's just like FITC. FITC, if combined with dextrin, can also be used as a dye to mark the lysosomal compartment. The main thing is the dextrin molecule. FITC or rhodamine is the fluorescence that is associated with the dextrin, which can be modified. Double fluorescence ring, red, blue and red, around a bead in microscopy indicates that the bead is external to the cell. The bead has not been phagocytosed. The bead is in the process of phagocytosis. The phagosome has fused with lysosome, forming a phagolysosome. The answer is D. If you just see a blue ring, then it means that it is been phagocytosed. It's in the phagosome state, not in a phagolysosome state. That's it for uh, unless there are any questions. Are there any questions? If there are any questions, I can stick around for a few minutes to answer them. Otherwise, that's the end of the class. Okay. If there are no more questions, thank you for listening. Thank you.